Hello, my name is Bryce McCourt, and this is my glycolysis, gluconeogenesis, and the role of aldolase gene expression presentation. So to begin with the introduction and student learning goals, so students can understand what is expected of them that they need to learn throughout these important concepts, is first, students will analyze the steps of the glycolysis and gluconeogenesis pathways. Students will understand how aldolase is involved in both the glycolysis and gluconeogenesis pathways. Students will know the different isoforms of aldolase and their specific expressions. And finally, students will evaluate how aldolase B expression is regulated by transcription factors. So as an overview of the glycolysis pathway, glycolysis means the splitting of sugar. So we're going to take a sugar molecule, which is known as glucose, C6H12O6. We're going to split it. Okay, it's going to be degraded in a series of enzyme-catalyzed reactions to yield something called pyruvate. And pyruvate is a molecule that contains 3 carbon atoms. And as a result of that, we want to make 2 pyruvate molecules for every molecule of glucose to conserve our mass in our chemical reactions, to balance our chemical equations. And because of that, we would need 2 pyruvate molecules to then add up to the 6 carbon atoms we originally started with the glucose molecule. And glycolysis is the very first step of cellular respiration where we are making ATP for energy. And it's going to occur in the cytosol of the cell. And it actually comes about with 2 phases. It has a preparatory phase and it has a payoff phase. And preparatory phase is what happens first. And this is the energy requiring steps. We need an input of energy for these steps to occur. There's the first priming reaction, there's a second priming reaction, and then there's the cleavage of a 6-carbon sugar phosphate to 2 3-carbon sugar phosphates. And during this phase, the phosphorylation of glucose comes about, and then it is converted to something called G3P, known as glyceraldehyde 3-phosphate. And then we have the payoff phase, and this is also known as the energy releasing steps. And this is whenever oxidation and phosphorylation steps occur. So the oxidation conversion of G3P comes about, and then it is converted to our 2 molecules of pyruvate that we need, and then the coupled formation of both ATP and NADH comes about as well. So we have the first ATP forming reaction, and then we have the second ATP forming reaction occurring, because we do make a net worth of 2 ATP molecules in the glycolysis process. So in regards to the individualized steps, glycolysis contains 10 steps altogether, and the very first 5 steps occur within that preparatory phase, within the energy requiring steps. And so step number 1 is whenever this glucose molecule is then converted to something called glucose 6-phosphate because of the phosphorylation of this glucose molecule, because a phosphate is added onto the glucose and is bonded to the 6th carbon atom. Next, glucose 6-phosphate is then converted to fructose 6-phosphate for the second step. Then, for the third step, fructose 6-phosphate is then converted to fructose 1,6-biphosphate. And then for the fourth step, this is whenever the enzyme aldolase comes about. And aldolase is important in here with the cleavage of the fructose 1,6-biphosphate. Aldolase plays a role in catalyzing the reversible aldol condensation reaction that occurs. And when this occurs, it makes dihydroxyacetone phosphate. And then for the 5th step of the reaction, this is whenever the G3P, or the glyceraldehyde 3-phosphate, comes about from the interconversion of the triose phosphates. And we create G3P at the end of the preparatory phase. Now next, in the steps of glycolysis, the very last 5 steps occur within the payoff phase. These are the energy releasing steps. So for step 6, we end up with G3P, the glyceraldehyde 3-phosphate, And what happens is this molecule is oxidized, and from the oxidation, we make 1,3-biphosphoglycerates. And everything here is going to be times 2 because we want to make 2 molecules of pyruvate at the end to conserve our mass. Then for step 7, a phosphoryl transfer is going to occur. So this phosphate here is going to break off, and it is then going to form the product 3-phosphoglycerates. And that phosphate group is on the 3rd carbon. For the 8th step, 3-phosphoglycerate will then be converted to 2-phosphoglycerate because the phosphate group has been changed to the 2nd carbon atom. Then for the 9th step, a dehydration is going to occur. Water is going to pull out of this reaction, so we're no longer going to have a water molecule, and then it is going to change into something called PEP, or known as phosphoenolpyruvate. And then for our 10th and final step of glycolysis, we're going to have a transfer of the phosphoryl group here. Okay, so the phosphate is going to come off. Okay, and then from there, we're going to create pyruvate. And again, we're going to create 2 molecules of pyruvate and have a net worth of 2 ATP molecules and 2 NADH molecules. Now, for the overview of our gluconeogenesis pathway. Gluconeogenesis means it's the new formation of a sugar. And gluconeogenesis is really the opposite of glycolysis. Glycolysis, we started out with glucose and we wanted to split it into pyruvate. But in gluconeogenesis, we start out with 2 molecules of pyruvate and we want to combine them together through enzyme catalyzed reactions to form sugar. Because living organisms need this method for synthesizing glucose from non-carbohydrate precursors. And we need this because our supply of glucose is not always exactly sufficient. In case we're athletes, in case we're going about in strenuous activities, we're going to need glucose and sugar for energy. And that is why we need a way to form new sugar. And this is regulated by insulin, glucagon, and cortisol. And it is specifically localized in both the mitochondria and in the liver. And we want to maintain blood glucose levels throughout a fasting state. So for the gluconeogenesis pathway, what happens is we start out with, and again, this is going to be times 2 because we have 2 molecules of pyruvate to conserve our mass. We're going to start out with pyruvate. Okay, we have all of these enzymes here that's going to catalyze these individual reactions. And what's going to happen is we're going to form the oxaloacetate first. And then, like I said before, this is going to be really the exact opposite of glycolysis, because after the 2nd step here, we're going to form the PEP, just like we did in glycolysis. We're going to make the phosphoenolpyruvate. And then from here, we're going to have an enzyme catalyzed reaction, and we're going to make 2-phosphoglycerates. Again, just like we did in glycolysis. Then it's going to switch. That phosphate group is going to go to that 3rd carbon for 3-phosphoglycerate. And then we're going to have a phosphate added on. We're going to have 1,3-biphosphoglycerate. And then we're going to make G3P, glyceraldehyde 3-phosphate. And then here's aldolase. Aldolase plays an important part in gluconeogenesis as well. For the formation of, after G3P is converted to the dihydroxy acetone phosphate, aldolase plays a role, and it is going to turn it into fructose 1,6-phosphate. And then from there, this is converted to just fructose 6-phosphate. And then it goes to glucose 6-phosphate. And then that phosphate group is going to break off. And then we're just going to get a glucose molecule. And we have formed the glucose that we needed. And then from there, the glucose can go throughout our blood into tissues that need it. And just as I said before, this is just an overview of really how glycolysis and gluconeogenesis are really just the exact opposites of each other. So for example, glycolysis is in the red here, and it's on the left side. And again, glycolysis starts out with a molecule of glucose, and then we're going to follow the red arrows, and we're going to go all the way down to the bottom. and we're going to form the 2 molecules of pyruvate. So we start out with glucose. These are all the enzymes, the specific individualized enzymes that catalyze each reaction to where we end up with pyruvate at the end. And aldolase is here in glycolysis to help us go from fructose 1,6-biphosphate to dihydroxyacetone phosphate, just like it did beforehand, and then to where we got G3P, the glyceraldehyde 3-phosphate. And if we would look at gluconeogenesis on the right side here in the blue, we would see that instead of the arrows going from the top and going to the bottom, gluconeogenesis is just the exact opposite. What it does is it starts at the bottom with the 2 molecules of pyruvate, and it goes all the way up. The blue arrows go from the bottom all the way up in just the individualized steps that we just went over. So here's the oxaloacetate that I just mentioned. Everything in blue here is those catalyzed reactions, the enzymes that play a part in those. This is where we have our PEP. Okay, this is our 2-phosphoglycerate, the 3-phosphoglycerate. It just goes up in the blue arrows, the exact opposite of glycolysis. And we also have the aldolase here as well in gluconeogenesis. Right here, whenever we're going from the G3P and we're going from the dihydroxyacetone phosphate, and we go up to the fructose 1,6-biphosphate. Aldolase is in the same spot, and you can see it across here in glycolysis and gluconeogenesis. So again, these are just the exact opposites of what they're wanting to do. Glycolysis was wanting to split the sugar molecule. Gluconeogenesis was wanting to take the pyruvate and form sugar from it. So now, talking about the isoforms of aldolase. Aldolase is a specific protein, and by specific protein, it's really an enzyme. Enzymes speed up chemical reactions. They're not necessarily consumed in chemical reactions, and they speed up the reactions by lowering the activation energy of the chemical reactions. And aldolase is just an example of an enzyme, multiple enzymes that we need. And this enzyme is particularly involved in glycolysis, gluconeogenesis, and also fructose metabolism. And there are different isoforms of aldolase. There's aldolase A, aldolase B, and aldolase C. And in particular, to go through each one of them, for aldolase A, it is the aldolase that is specifically in glycolysis. And it is the glycolic enzyme that catalyzes the cleavage of fructose 1,6-biphosphate to the glycerol 3-phosphate, or glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate that we just saw in the diagram. And in particular, aldolase A is very, very abundant in our muscles. And it can also be expressed as a mutation that can result in a very specific anemia that is called non-spherocytic hemolytic anemia whenever there is a mutation on the aldolase A gene. And that specific mutation is caused by an amino acid substitution where this amino acid and this amino acid switch places. And it destabilizes the structure of the enzyme because if we change the DNA and change an amino acid in an enzyme, in a protein, we change the structure. And when we change the structure, we also change the function because structure equals function. And so because of that, it's going to completely change that enzyme and could destabilize it. And this aldolase is actually the most studied isoform of aldolase of the whole entire family of aldolase. But even though it's the most studied, we still don't necessarily understand its whole catalytic mechanism. We still don't understand everything about it. Now, with aldolase B plays a very crucial role in the fructose metabolism and in gluconeogenesis. So aldolase B is the specific aldolase that we see in gluconeogenesis. And this expression is very restricted to the liver, to the kidneys, and to the small intestine. That's where we see aldolase B. And it could also be a mutation on the Aldolase B gene, and it could cause a recessive disease known as hereditary fructose intolerance. And its greatest catalytic efficiency is in the liver, because the liver is actually where gluconeogenesis takes place. And then for Aldolase C, it is found in the brain and in other neurological tissues, specifically in vertebrates. And it is also found in tissues of just constant metabolic activity. And it is also present in fetal and cancer cells, and its specific functional role, physiological role of Aldolase C, it's still not necessarily understood. It's very unknown still to this day. And it is composed of catalytic properties that are immediate to those of Aldolase A and B. And whenever you compare, when scientists have compared the nucleotide sequences of Aldolase A, B, and C, the comparisons have showed that A and C resemble each other, but not necessarily with B. So Aldolase A and C are very closely related to each other, not necessarily with B though. So if we'll look here at the picture, this is just a diagram that I found of the different isoforms of aldolase. Again, this is Aldolase B that we see in the glycolysis reactions, abundant in our muscles. B is in gluconeogenesis. This is in our liver, kidneys, our small intestines. And C is in neurological tissues in the brain. It is the one that closely resembles A, especially in these black marks you can see. It does resemble more of the A than it does with B. And also in regards to nucleotide sequences as well. So whenever we talk about Aldolase B expression and regulation, in particular, again, Aldolase B is specifically the isoform of the aldolase family that is in gluconeogenesis, where we're wanting to form glucose out of the two molecules of pyruvate. There are tissue-specific expressions, in particular to the liver. That is where most of the catalytic efficiency of Aldolase B is. It is determined at the level of transcription initiation. So this is when we are going to begin discussing the transcription factors, how they could regulate the Aldolase B expression. And there are several liver-specific or liver-enriched DNA binding proteins that are factors, and they control liver-specific transcription. And so, for example, one of them is HNF1, hepatocyte nuclear factor 1, HNF3, hepatocyte nuclear factor 3, and there's also one where it has a V before it, and it's VHNF1. That is just, for example, a variant of the hepatocyte nuclear factor 1, and we'll see that here in just a bit. Another one is C-EBP. This is an enhancer binding protein. And then a related protein such as DBP, this is albumin D element binding protein specifically. So if we'll look here at these graphs, so these studies went about specifically for the Aldolase B gene expression, about how these specific transcription factors regulated it. And so here on the y-axis, we have the catalytic activity, okay, on both of the graphs. But this one in particular, we're looking at the expression vectors here and how each transcription factor goes about in regards to catalytic activity. And so first, what we see is that the ones that have the highest, that are the ones here, this one, and this one here. This first one is HNF1. And the second one is DBP. And these two are, for example, in the liver cells, they are transactivators. And by transactivators, what they do is they bind to a specific promoter region of the DNA, and then from there, that gene will be expressed. So what they do is they have very high catalytic activity. They have the highest catalytic activity out of them all. Specifically, HNF1 has the highest. in that case, because they are what is responsible for expressing a gene. Okay, so that's how they regulate aldolase B. And now, this one here, this is the VHNF1 that I explained earlier. This is a variant of the HNF1. It is actually very hard to see because it's very low here on the diagram. It has a very low catalytic activity. And then the next one is the CEBP. It's this one right here that starts and then it stays there on that x-axis and only just increases a little bit. So both of them have very low catalytic activity. So they are very weak activators. They are definitely not as strong in catalytic activity as HNF1 or DVP that are the trans activators. So they are the weak activators with low catalytic activity. And then the final one here, it is HNF3. And it is very, very low. It is very hard to see. It stays here on that x-axis and baselines, and it does not have a very high catalytic activity. And so that tells us from this graph, in regards to HNF3 and catalytic activity, it tells us that it is very inactive when it comes to regulating the aldolase B gene and its expression. So it's very inactive in regards to that. Now, if we'll switch to this graph here tells us catalytic activity compared to if we're going to combine different transcription factors. And this was done with the PCR. So this one here, this 1, 2, 3, 4, these first 4 here are what they look like just by themselves. So this, for example, is the DBP by itself, CEBP by itself. This is HNF1 by itself, HNF3 by itself. And it is very consistent with the graph over here. HNF1, DBP have the highest ones. And out of the highest, out of the 4, it's HNF1. Now, if we'll look here, this is DBP and HNF1. If we put those two transcription factors together, they are the highest out of anything, even out of the individualized categories. So they are the highest ones. Again, they are those trans activators. They are the ones that, in regards to the gene, they're expressing it. Okay, now, if we look at anything that has HNF3 in it, which is the one that was very inactive compared to everything else, HNF3, if we would combine it with DEP, it is very, very low. Very, very low here. So this is what DEP was by itself. And this is what it is with HNF3. Okay, so it goes down very, very low. Now, if we would look at HNF3 and HNF1, it goes down very low as well because HNF1 by itself was here. If we mix it with HNF3, which starts out here just by itself because it's very inactive, it is a dramatic decrease. So that is telling us that not only is HNF3 very inactive in catalytic activity, it is actually going to inhibit, okay? It's actually going to competitively inhibit, specifically with HNF1. Because HNF1 was very high by itself when we combine it with HNF3, and it is not. So it is an inhibitor, okay? And it doesn't necessarily express the gene. It's going to inhibit if it binds, okay? Just a few examples there. Now, in regards more to aldolase B expression regulation, if we would switch and look at kidneys, the proximal tubules from the kidneys, they were characterized by the absence of HNF3 and the presence of higher ratio of HNF1 and HNF1A transcripts. So again, this goes back to our graphs. HNF3 was very inactive. HNF3 was very inactive, but HNF1, and in regards here, they broke it down to A and B. HNF1 was a trans activator. It was the highest one out of the transcription factors we're looking at. HNF3 is very inactive. And there's evidence showing that the HNF3 factor plays a similar negative role, and it competitively binds close to or on the HNF1 site, which is exactly what our graph showed us as well. We combined HNF3 with HNF1, and it competitively inhibited it. And it went from very high catalytic activity to very, very low catalytic activity, which is what our graph showed us. So again, HNF3 is the inhibitor. It is very inactive in regards to catalytic activity. HNF1 was the highest in catalytic activity. but if it is bound with H and F3, it's going to competitively inhibit with it. So as a review, glycolysis is composed of a total of 10 steps that results in the splitting of glucose molecule to the 2 molecules of pyruvate. Gluconeogenesis is composed of 11 steps if you want to take into consideration oxaloacetate at the beginning. It's the 11 steps, and this results in the formation of glucose when we start out with the 2 molecules of pyruvate. Again, they're just exact opposites of each other. Aldolase plays a role as a catalyst in both the glycolysis and gluconeogenesis pathways. When we talk about that aldol condensation reaction, aldolase can be expressed as A, B, and C in regards to the aldolase family. And in particular, aldolase B can be regulated through transactivators, repressors, or inhibitors that we just talked about. And these are my references. Thank you very much.